Hello, good morning, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here to present my new lecture on the cell organs, structure, and functions. So, friend, this is very important topic for all the life sciences exams. And in the previous lecture, we have discussed about the cell, structure of cells, membrane structure, cell wall structure, and the cell division process. And in this lecture, we will discuss about the cell organelle, nucleus, endoplasmic reticulum, Golgi body, protein targeting, oleosomes, microbody, chloroplast, mitochondria, and cytoskeleton. So, friends, let's start our lecture with the cell organelle. So, what is the cell organelles? So, cell organelles are the permanent and metabolically active living structures of cytoplasm is called as a organelles. And these organelles are classified into three main categories based on how they are arranged. So first one is the endomembrane system, and second one is the independently dividing organelles derived from the endomembrane system, and third one is the independently dividing semi-autonomous organelles. So friends, in the first system, we include the group of membranes or organelles like endoplasmic reticulum, nuclear envelope, Golgi apparatus, vacuoles, and endosomes. So friends, these all together and play a central role in protein secretory process, membrane recycling, and the cell cycle. And second one is the group which include oil bodies, peroxisomes, and glyoxisomes, which function in lipid storage and carbon metabolism. And third one is the include plastid and mitochondria. So these are semi-autonomous organelles and functions in lipid storage and carbon metabolism. So friends, based on their arise, the cell organelles are into three main category. First one is the endomembrane system. Second one is the organelles which are derived from the endomembrane system. And third one is the independently dividing and semi-autonomous organelles. So friends, this is a structure of reference plant cells. And you know that a plant cells have several cell organelles such as cell wall, cell membrane, Golgi apparatus, chloroplast, vacuolar membrane or vacuoles, graphite crystal, and Druce crystal. Friends, these are the calcium oxalate crystal which act in the defense against herbivores. Mitochondria, cytoplasm, then amyloplast, then vacuole, rough endoplasmic reticulum, nucleus, smooth endoplasmic reticulum. Its work in the lipid biosynthesis, and rough endoplasmic reticulum in protein biosynthesis, ribosomes in protein biosynthesis, and Golgi vesicles. So friends, these are the important organelles of plant cells. So let's start with the nucleus. So friends, you know that nucleus, it is discovered by the Howard Brown in 1831 in orchids and named as a areola. And the nucleus can be stained by any basic dyes such as acetocaramines. And the nucleus is the largest cell organelle in the animal cells. But in the case of plant cells, the largest cell organelle is the chloroplast. And also, friends, nucleus is the first organelle, first organ to be discovered so far. Okay, friends, and the nucleus is derived from the Latin word that is nucleus, which means the kernel or seed, because it is the central part of the central organ of the cells. It contains the genetic information and primarily responsible for the heredity, which cause it contains DNA, metabolism, growth and differentiation of the cells. Study of the nucleus is called as a karyoology. Generally, eukaryotic cells contain at least one nucleus or many. It can be one or many. Generally, more active cells have more nucleus. And the nucleus is absent in mature phloem cells, sheep tube elements, and mature RBC of mammals. Also, the collectively genes for heredity, growth, and metabolism and their intervening sequence are referred to as nuclear genome. The size of nuclear genome is vary in 1.2 into 10 to the power 8 base pair and adopsis to 1.2 into 10 to the power base pair in lily fritillaria. So friends, this is the structure of nucleus, in which we have nuclear envelope which contain double membrane. So nucleus have double membrane, the outer, and second one is the inner membrane. This is the outer membrane and this is the inner membrane. And also it have dense nucleus, then nucleoplasm. Also it have DNA bind chromosome, that is chromatin, heterochromatin and euchromatin. Then we have ribosomes on the surface and we also have nuclear core. So we will discuss about the structure of. So let's start with nuclear envelope. So friends, as you know that nucleus have double membrane structure. Is outer membrane and inner membrane. So this outer membrane cover the inner membrane and it's also continuous with the adjacent ER, means it connected with the endoplasmic reticulum and stuffed with the ribosomes. So ribosomes found on the outer membranes, while the inner membrane surround the nuclear content, that is nucleoplasm, and also provide the shape to the nucleus. So this is the function of outer and inner membranes. Also, the nuclear pores form selective channels across both membranes connecting the nucleoplasm with cytoplasm. So friends, the both outer and inner membranes form a structure that is called nuclear membrane pores. It's from the nuclear pore. So the size, what is the size of this nuclear pore? It is near to 105 nanometers. Also, there are some proteins are present that is called nucleoporins, and there are more than hundreds of nucleoporins are present. So these nucleoporins are arranged octagonally and form a 105 nanometer nuclear vortex. So friends, these act as a supramolecular sieve. So the nuclear pore complex act as a supramolecular sieve. So they are a special amino acid sequence called as nuclear localized signals is required for a protein to enter the nucleus, and the perimeter space, the space between the two nuclear membrane, outer and inner membrane, is 10.50 nanometer. So friends, in this slide we have discussed that there are two membranes of nuclear membrane, that is outer and inner membranes, and the outer membrane is connected with the endoplasmic reticulum, and inner membrane provide the shape to the nucleus. And some proteins, that is nucleoporins, which are formed a nuclear pore between these two membranes, and which is required for the protein to enter. And there is one that is called NLS, nuclear localized signal, which is required for protein to enter into the nucleus. Also, there are some proteins which control the in and out the nucleus. This is called as a impartins and expartins. These proteins also have role in the movement of molecules through the. Also, the larger molecules, more than this 40 nanometer, cannot be moved through this force. Now nucleoplasm. So nucleoplasm or nuclear shape is a round substance of nucleotide, nucleosides, ATP, protein, and enzymes of RNA and DNA polymerase, endonuclease and minerals. So friends, this is a nuclear shape which contain all the nucleosides, your ATP proteins, enzymes, RNA, DNA polymerase, and everything is found in the nucleoplasts. Also, the chromatin net and nucleus are embedded in the nucleoplasm, and this nucleoplasm provide the site for the process of transcription. And what is chromatin nets? So interphase nucleus has a loose and indistinct network of nucleoprotein fibers, which is called as a chromatin, and this chromatin net is embedded in the nucleoplasm, and they made up of this chromatin made up of DNA and stone proteins. And the term, this chromatin name given by the W. Fleming in 1882. And there are two types of this chromatin. This is euchromatin. It is also called as a true protein. And second one is the heterochromatin, which is also again two types, constitutive and facultative heterochromatin. And these proteins are interconvertible to each other according to the situations. So friends, generally in the cells, only 10% of euchromatin are present and 10% of heterochromatids are present, and the other 80% are in intermediate form. Which can be converted according to the situations. And last is the nucleus. So nucleus is the dense structure and the site of ribosome synthesis, includes a portion of chromosome where rRNA is inside the ribosomal RNA, and this called as a nuclear organized regime. So what are the functions of nucleus? It contains the genetic information, it transmits the genetic information, also it have role in cell division, and it's control the metabolism and genetic variation. So friend, this is the important structure about the structure and function of the nucleus. Now let's move to endoplasmic reticulum. So the important transport. So friends, this is the important transporting system of the cells or eukaryotic cells, because it is absent in prokaryotic cells, and name given by the Porter 1953. So this endoplasmic reticulum have three components. One is the cisternae, second is the vesicles, and third one is the tubules. So cisternae are long, flattened, and unbranched unit arranged in a stacks. Second one is the vesicles, which are oval membranes bound structures, and third one is the tubules, which are irregular in shape and often branched tubes bound by a membrane, and these tubes can be free or associated with the cisternae. And this a very large part of the endoplasmic reticulum resides in the outer layer of cytoplasm, which called as the cell cortex, but it also forms tubules bundle that transverse the cell through the transvascular strands. And the polygonal networks, this tubule network is physically connected to the plasma membrane at some of the points of intersection, which is called as a polygonal network. And there are one protein that is called reticulones. This protein helps in the transition of tubular and cisternae form, whereas the actomycin cytoskeleton activates endoplasmic reticulum tubules rearrangement, cisternae formation, and the flow of protein. So this reticulum protein help in the transition of tubules and cisternae forms, and actinomycin help in the tubules rearrangements and cisternae formation and flow of protein. And the region of endoplasmic reticulum that has many wound ribosomes, which called as. So friends, you know there are two types of endoplasmic reticulum. First one is the rough endoplasmic reticulum, in which the ribosome are stuffed and have important role in the protein synthesis, while the without ribosomes is called as a smooth endoplasmic reticulum, which have role in the synthesis of lipids. And RER shape was like a flattened small sac, and is smooth endoplasmic reticulum being a tubular, and mostly flowering glands which are producing nectars contain much of SER. So in the flowering gland, which endoplasmic reticulum are dominating, that is smooth endoplasmic reticulum. And this endoplasmic reticulum provides the membrane building blocks, because it's inside the phospholipids, and protein cargo, means in the it have important role in the protein trafficking, means transfer of protein or targeting proteins for the other compartments in the endomembrane pathway. And the endoplasmic reticulum is the major source of membrane phospholipid used to construct the entire endomembrane system. And the intrinsic sidedness or asymmetry to the membrane bilayer is because the enzymes that initiate phospholipid synthesis on the endoplasmic reticulum add new phospholipid to the more to the cytosolic leaf plate of the bilayer. So here two enzymes that is called cleavage and clopage. These two enzymes have role in flip flop movement of cell by proteins. Okay, friends. And the cell fission and fusion cycles form the basis for the growth and division of the endomembrane system. And here some two protein that are S and A R E S and this RAPS protein. These two proteins regulates the cell fission and fusion cycle, which is basis for growth and division of the endomembranes organelles. And these are the modification of endoplasmic reticulum. So first one is the sarcoplasmic reticulum. So sarcoplasmic reticulum in the skeletal and cardiac muscles function as a storing of calcium, iron, and ergostoplasm. Ribosomes are accumulated on the small parallel cisternae of endoplasmic reticulum, then called ergostoplasm, and generally it is found in nerve cells or mucous body. So friends, this is mainly in animal cells. And third one is the myeloid body. This is specialized SER, which is found in pigmented epithelial cells of retina, and the myeloid body is a light sensitive structure involved in pigment migration. And fourth one is the microsomes, which is associated with the ribosome particle and obtained by the fragmentation and high speed centrifugation of cells. And friend, this does not exist in the living cells. So generally this question can be asked in MCQ that which organ is not present in the living cell, that is microsomes. So what are the important functions of endoplasmic reticulum? So with microtubules form, it forms the endoskeleton of the cells, and it also intercellular exchange of material. Also role have protein synthesis, RER, lipid biosynthesis, cellular metabolism, formation of nuclear membranes, formation of lysosomes, Golgi body, and some also microbodies, also some detoxification, and provide precursor of secretory material to the Golgi. Now next is the Golgi body. So Camellio Golgi in 1897, or in plants it is also called as a dictyosomes, discovered or named this Golgi body. This is a polarized stacks of cisternae with the flatter cisternae occurring on the cis side or forming phase, which accept tubules and further vesicles from the endoplasmic reticulum. So friends, here are two sides of this Golgi body. One is the cis phase and second one is the trans phase. So cis phase is toward the endoplasmic reticulum, which accept the tubules from the endoplasmic reticulums, or this trans phase budding of the vesicles. And the opposite maturity phase of the trans side has flattened thinner cisternae and include a tubular network called trans Golgi networks, and this cis phase is called cis Golgi network, and this is called as trans Golgi network. And this secretory vesicles may bud out from the trans cisternae or trans Golgi networks. So vesicles, which are these are the vesicles transported from the endoplasmic reticulum, moved from cis phase to trans phase, and then later bud off from the transphase. And this Golgi body can divided by the splitting process and regulate their capacity for secretion during growth and differentiation. So, delivery of membranes and its content to the Golgi body from endoplasmic reticulum occur at specialized endoplasmic reticulum exit sites determined by the CO2 proteins. So, friends, there are one that uh, so at the cis side of Golgi body, there is hope second protein which accept the delivery of membrane and its content. And at the endoplasmic reticulum, there is a exit site which is called as endoplasmic reticulum exit site, where this membrane and its content comes out and transport out of the endoplasmic reticulum to the Golgi. Within the Golgi cis to trans phase means contents come to cis to trans, that is called anterograde movement, and later on this transport the material to the plasma membrane and other organelles. This is called as a anterograde movement, while the opposite of this is called as a retrograde movement, and in this COP1 protein are involved. And the function of Golgi body is the cell secretion, means secretion of macromolecules, director of macromolecules traffic in a cell or middleman of the cells, because it transport the macromolecules, synthesis of cell wall materials like polysaccharide synthesis. It also have role in cell plate formation, phagmoplast during the shell wall formation of acrosomes during spermatogenesis process, and formation of lysosomes, which is joined by the Golgi body and endoplasmic reticulum. Now, next is protein sorting pathway. So, friends, we know that the proteins or transcription process occurs in the yes process, and here that newly synthesized proteins which are destined for the proper cellular organ, means this is called as a protein targeting or sorting, means the delivery of newly synthesized protein to their proper cellular destination, means where they want to need or where they are need. It is called as a protein targeting or sorting, and this can occur by two ways. First one is the signal based targeting, and second one is the vesicle based trafficking. So friends, in this first process, this is the newly synthesized proteins, which this is the mRNA or this is the nascent polypeptides, and it's moved from nucleus to cytoplasm, and where there are some targeting sequence which are attached to these proteins and targeted for the peroxisomes, chloroplast and mitochondria by the help of addition of this targeting sequence. And the second process in which these proteins, transcription proteins, are moved to the endoplasmic reticulum, then endoplasmic reticulum to it's moved to Golgi vesicles, and then moved to different organelles. So this is the process of. So you know that first process is the replication, which occurs in nucleus, where DNA to form DNA. Then there will be a transcription process, transcription, where formation of DNA to synthesis of RNA. And different types of RNA like mRNA, rRNA, tRNA, messenger RNA, ribosomal RNA, tRNA are synthesized in the nucleus. And now there is a processing of this transcript unit, transcripts, where there is a addition of 5 cap. At 5 end there is addition of cap, and at third end there is addition of tail. Okay friends, now this polypeptide move to the cytoplasm, transcript move to the then protein synthesis on the ribosomes, and then after the synthesis of proteins there are binding of some signal recognition particles, that is also called SRPs, which are attached to the signal peptides. So in this transcripts there are 15 to 30 amino acids which act as a signal peptide. So where is the attachment of this SRP particle? After addition of this SRP particles, there will be a transporting channel, that is called translocons, which are present on endoplasmic reticulum, where this polypeptides move through the endoplasmic reticulum and they are binding of ribosomes. Then these signal peptides and cleaved out, and then the process of post translation modification, in which addition of carbohydrates, and this then later on this post translates product move to the Golgi body and then move to the cis side to the trans side, is anterograde movement, then what of and destined for the organelles. So this is the second process of protein trapping. Now come to vacuoles. So friends, this is the structure of vacuoles. So membrane of this vacuoles is called as a chloroplast, and it have cell sap in the central vacuoles. So a membrane enclosed compartment without the cytoplasm. In this organ, instead of cell sap, instead of cytoplasm, it has cell sap which contain water and solutes. And the increase in the volume of plant cells during growth phase take place primarily through an increase in the volume of vacuolar cells. So the increase in cell during the maturity mainly because of the big vacuoles, and the meristematic cells, they have no central vacuoles, or if they have central vacuoles, then vacuoles are smaller vacuoles, not big vacuoles. So mature cells have big vacuoles. And the vacuolar membrane, this is called tonoplast, contain lipid and proteins, and initially synthesized in the endoplasmic reticulum. It also regulates the cell expansion. The vacuoles can also serve as a storage compartment for the plant's secondary metabolites involved in plant defense against herbivores and biotic stress. Also, inorganic iron, sugars, organic acids, pigments can also accumulate in the vacuoles. Also, toxins which are produced during the metabolic process also accumulate in the vacuoles. That's why it is called as a storehouse of the cell. And vacuole play important role in protein turnover. In the case of lytic vacuoles that accumulate in aged leaf, which release a hydrolytic enzymes which degrade the cellular constituent during senescence. So during senescence process, which type of vacuoles, that is lytic vacuoles, are activated and hydrolyze the your carbohydrates and other macromolecules which cause for the senescence. In some plant, lytic vacuoles are derived directly from endoplasmic reticulum bypassing the Golgi body. And the delivery of Golgi derived vesicles to the vacuoles is indirect, means without incorporating the Golgi body, directly transferred to the this. Then there is the multivascular body, MBB, is a post-vacuolar compartment that function in the degradation of vacuoles and their membranes. A pre-vacuolar compartment which also serve as a sorting organelle for membrane endocytosis from the plasma membranes. So now come to seconds, that is independently dividing organelles derived from the endomembrane system. So first one is the oleosomes. So friends, oleosomes represent the unique. Why it is unique? Because it have half unit membrane, that is phospholipid monolayer instead of phospholipid bilayer and derived from the endoplasmic reticulum. And the phospholipid in the unit membrane system is oriented with its polar head grouped toward the aqueous phase of the cytosols, and the hydrophobic fatty acid tails facing towards the lumen, which is dissolved in the stored lipid. So this is the structure of oleosomes. And oil bodies are initially formed as a region of differentiation with the endoplasmic reticulum, means endoplasmic reticulum involved in the synthesis of these oil bodies, and the nature of storage products are triglyceride, PAG, which dictate that this storage organelle will have hydrophobic lumens. As triglyceride storage, it's appeared to be initially deposited in the hydrophobic region between the outer and inner leaflet of endoplasmic reticulum membrane. When the oil body separates from the endoplasmic reticulum, it contains a single outer leaflet of phospholipid containing a special protein that is called oleosin. So this is the protein. And oleosins are synthesized on cytosolic polymers which are inserted into the oil bodies, and the protein consists of central hairpin-like hydrophobic regions which insert itself inside the oil containing lumens. And when oil body break down during seed germination, they are associated with other organelles that contain enzyme for lipid oxidation such as glyoxisomes. So these are during the germination, these oleosomes are attached with the glyoxisomes, and they store large quantity of oils in the phase of seed development. So mainly this is the oil body which synthesizes during the seed development process. Then microbodies. So this is the structure of microbody where the plasma membrane, and this is the crystalline core, and there is a lipid bilayer. And this is a class of specialized organelles covered by the single membranes, not double membranes. And peroxisomes and glyoxisomes are the microbodies which are specialized for the oxidation of fatty acid and the metabolism of glyoxylates, A2-carbon aldehydes. And these microbodies lay DNA and are closely associated with other organelles with which they are share intermediary metabolites. The glyoxisomes is associated with mitochondria and oil bodies, while the peroxisome is associated with mitochondria and chloroplast. Means Peroxisomes have important role in during the photo respiration and associated with the mitochondria and chloroplast, while glyoxisomes uh, associated with mitochondria and oleosomes during the seed germinations. Okay. And they are also interconvertible, means peroxisomes developed directly from glyoxisomes, at least in the greening cotyledons, means at non-greening cotyledons they contain glyoxisomes, but after greening there will be existence only peroxisomes, means there is no glyoxisomes. And intermediate stage glyoxisomes are converted to peroxisome during the greening process. In peroxisomes, glycolate is oxidized to acid aldehyde glycolate. During this conversion, there is formation of H2O2, and most of the protein of peroxisome is catalase. So peroxisome is rich in catalase protein, which break down the hydrogen peroxide into the water. And catalase often so abound in peroxisomes that it forms the crystalline array of the proteins. And most protein enter in the peroxisomes from the cytosols post translate by the means of targeting signal consist of serine, lysine and leucine. Right? These are the signal sequence of the targeting specific targeting signals for the peroxisomes. Now friends, come to independently dividing semi-autonomous organelle, that is called mitochondria. So this is the structure of mitochondria. So you know that mitochondria is derived from the prokaryotic cells. Prokaryotic cells. It have mitochondrial DNA, that is bacterial type 70S. It have ribosomes. It have matrix granules. It have lamina, history in folding of inner membranes, and the outer membrane which have porins, and the perimitochondrial space, and the complex of ATP synthesis. So this is the basic structure of mitochondria. It is discovered by the C. Vanda and named as a chondriomes. This is called as a population of all mitochondria in a cells, because in a cells there are more than one or many mitochondria, and the active cells there are higher number of mitochondria, and in the senescent cells there is less number of mitochondria. And mitochondria are the primary site of cellular respiration, a method in which the energy released from sugar metabolism and used for the synthesis of ATP and ADP. We will discuss later on in some next lectures. And mitochondria are highly dynamic structure that undergo both fission and fusion, and this fusion process can result in the long tube-like structure that may branch to the mitochondrial network. And this is a double unit membranes, and without membrane the mitochondria is called as a myoblast and present in the metabolically active cells. The inner membrane is folded into the several finger-like crystals, and regardless of shape, all mitochondria consist of a smooth outer membrane and a highly convoluted inner membrane. Means the outer membrane is smooth and the inner membrane is a convoluted, means folded. And the inner membrane contains protein pumping, that is ATP synthesis, that uses a proton gradients to synthesize the ATP for a cells. This is also we will discuss in the photosynthesis and respiration. And the protein gradients is generated through the cooperation of electron transports, that is called as a electron transport chains, which is embedded in and peripheral to the inner membrane. And the phosphatidyl choline, phosphatidyl ethanolamines, diphosphatidylglycerols enzymes mainly present in the mitochondrial membrane. And the inner membrane have pinhead particles that is called as a oxysomes or elementary particles F1, F0, which are 10 to power 2, 10 to power 6, and also these are called as a French dense Moran particles. F1 is concerned with the oxidative phosphorylation, and ribosome is 70S type, which is prokaryote types, and single, double stranded, and circular knotted DNA. And the mitochondrial DNA is rich in the GC, and this is semi-autonomous because it's code for some protein, not for all. The function, this is also called as a powerhouse of the cells or ATP mill in the cells. Also it is called as a cell within cells because endosymbiotic theory, because mitochondria is the prokaryotic cells under the eukaryotic cells. Cell furnace or storage batteries, and also it is also known as most busy and active organs in the cells. So friends, now come to the chloroplasts. So this is the structure of chloroplasts. So in the chloroplast, we also have DNA, we also have ribosomes, starch granules, stroma, granathylakoids, thylakoids, humans, chloroplast inner and outer membranes, inner or perichlor, this inner membrane space, and the granum. So this is the common structure of chloroplast in the plant cells. These have double membrane and semi-autonomous nature. Means it also have DNA, but it code for some protein, not for all. And this is rich in glycosylglycerides, not phospholipids. Membrane of chloroplast contain chlorophyll and associate protein, and this is the site of photosynthesis. In addition to the inner and outer envelope membrane, chloroplast possesses a third system of membrane that is called thylakoid system. And a stalk of thylakoid form a granum. This is the granum. And protein, pigment, chlorophyll, and carotenoids that function in the photochemical process of are embedded in thylakoid, and these thylakoid membranes have these pigments, chlorophyll and carotenoids. And the fluid compartments surrounding the thylakoids called as a stroma, which is similar to the matrix of the mitochondria, and these adjacent grana are connected by a unstacked membrane that is called as a stroma lamellae. And ATP synthesis of the chloroplast is situated in the thylakoid membrane. And chromoplast contain a high quantity of carotenoids, which is mainly in the ripened fruits. And leucoplast is a non-pigmented and amyloplast, which function as a starch storage during the harvesting. All type of plastid have common origin from the proplastid, sac-like and non-laminar structure. In dark, reticulum contain a semi-crystalline tubular array of membranes known as prolamellar bodies. And the both mitochondria and plastid divided by the process of fission. This is the bacterial cell process of types of bacterial division or prokaryote divisions consistent with their origin. And protrusion of outer and inner membranes occurs in both mitochondria and chloroplast. In chloroplast, this protocol called stromules, which contain stoma but no thylakoid. In mitochondria, these are called as a matricules, and they work as a connecting the organelle from which they arise, and also therefore exchange the materials. So stromules and matricules work as the exchanging materials. And organelles can move or motorized by a plant myosin that moves along actin. In plant, amyloplast act as a gravity sensor in root. This we will discuss in the oxygen hormone. Each chloroplast contains about 20 to 100 granum and quantasomes located on the thylakoid membranes, which are numbered 250 to 400. And rubisco contain the major 60% protein of chloroplast. So rubisco is the major protein and ribosomes are 70x types. And its function in photosynthesis, balancing of O2CO2, cytoplasmic inheritance, greenery to earth, and storage vitamin K and C. Now come to ribosomes. So this is the smallest organ of the cells and discovered by the Palade 1953. That's why it is called as a Palade particles. And this is the structure of ribosomes. And it is also called as a engine of the cells. And it is made up of RNA and proteins. And this organ is without organelle. So this question also can be asked. Membraneless organelle in the cells is ribosomes. It is also as a protein factory of cells. And this is also a smallest cell, organelles. It is also called as a organelles within organelles. And in eukaryotic cells, they are 80S ribosomes. That is made up of 60 and 40 types. While and contain 40% ribosomal RNA and 60% protein. And while in the prokaryotes, it is 50S plus 30S. So large subunit is 50S and small subunit is 30S. And the 60% RNA, rRNA and 40% proteins. So this 50S divided F2 subunit, that 5S and 23RS sRNA, and 30S have one subunit, that is 16R RNA. And this in eukaryotes, as large subunit have 60 and small is 40S, and 60 are divided into 3 subunits, that is 5S, 20 or 26 or 20S RNA, and 5 sRNA. So you can see 5S RNA are common in the both prokaryotes and eukaryotes, and this shorter, that is small subunit have is 18S RNA. And this S refer to the Swedberg unit. This is the name of scientist. And the magnesium ion is essential for the binding of 2 subunit of ribosomes, and larger subunit of ribosomes for the peptidal transferase activity during protein synthesis. And now come to the cytoskeleton system. So the cytoskeleton contain an array of fibrous protein collectively called as a cytoskeleton. And there are 3 types, that microtubules, then microfilaments, and third one is the intermediate filaments. These are not present in the plants. So microtubules are the polymers of tubulins, which is 25 nanometer diameters, and made up of this tubulin protein is made up of alpha and beta tubulins, while the microfilaments is 7 nanometer diameters and polymers of actin polymer, while intermediates is 10 nanometer diameters. And this cytoskeleton provides strength and rigidity to cells, movement of structure within the cells, and also have role during the cell divisions. And some plant motor proteins such as myosin, kinesins, and dyneins that move in a direction or energy dependent manner along the cytoskeleton to translocate a variety of molecules or cellular structure. So these proteins act with this cytoskeleton proteins. And raw family small GTPase are important regulator of actin binding proteins and actin organization. While the identification of plant microtubules, associate protein MAPS directly bind to the microtubules and control the microtubule organization. and this cytoskeleton is important for the movement of the cells. So friends, in this lecture we have discussed about the cell organelles, and this topic is very important for the generally MCQ questions. Thank you very much.